All right, we're good to go. Thanks. Okay, I might have gotten a little carried away here. I'll cruise through this real fast, but it's a really exciting day for Maddie and all of us in the Wetland Ecology and Restoration Lab. So I want to tell you a few things about Maddie. It's a few surprising things, and maybe some of you don't know. And I always like to go back and kind of look at early emails with people on people's resumes when they first applied, because it's always very insightful. So it may appear that Maddie is firmly rooted in the West, in the wildlands, and the deserts, and the wetlands, but actually she's Florida, and she is a very passionate. They're going talking about Gainesville. You'll expect at least an hour-long conversation about her favorite restaurants and bars, as we witnessed recently. So she got her BS in interdisciplinary studies from University of Florida, a concentration in marine sciences, and a minor in art history, which I think adds a layer of depth that I didn't quite realize or appreciate. Some unsurprising things. She came to us with tons of field experience in seeds, restoration, wetlands, and riparian habitat. So she worked with AmeriCorps as a habitat improvement specialist, in Seeds of Success as a field technician with the Chicago Botanic Garden. Many of the folks in my lab have gone through that program. She was an ecological monitoring field technician with the Great Basin Institute. And one thing of note, I was looking back at a reference from that position, and her former advisor boss said she's so good at writing that I had her prepare nearly all of our reports. And in fact, I still use her as a resource to review my resumes. So keep that in mind. She also was a biological science technician with the Forest Service. But if you go a little bit further back in her resume, say to the winter of 2018, 2019, 2020, she was the build crew for Ice Castles, LLC in New Hampshire. So, you know, when I see these things on students' resumes, I think, okay, this is going to be really interesting and perhaps very telling about who this person might be. So her former boss said, from early on in her first season, Maddie could keep up with even our most seasoned builders. Not only could she churn out structures at a prodigious rate, but these structures were invariably safe and easy for the next builder to work on. She demonstrated a talent for finding creative solutions to problems. That is basically what a master's degree is all about, finding creative solutions. And she also created an effective means for tracking our redundant backup systems. And the final thing that I noticed from this reference was his comment about her unwavering work ethic and her moral compass. So all of these things really, Maddie has demonstrated. It's Valentine's Day, so I decided to compile a list of a few things from some of these references that also speak to me in terms of who Maddie is. Exceptional organizational skills, professionalism, dry sense of humor, willingness to learn new things, intelligence, determined, wonderful, genuine, honest, self-starter, and motivated. Happy Valentine's Day. So all of these qualities are essential for her success in her master's degree, and particularly for this really interesting research project that she's going to tell you about today. There were lots of field challenges and greenhouse and mesocosm challenges, as there always are, but she has a really interesting data set that I think will be very impactful in terms of Great Salt Lake restoration and wetland restoration in the broader Intermountain West. So I'm going to turn it over to Maddie now. I'll leave this mic on.
That's how it goes.
It's far not hanging and did take a longer.
Go with that. Okay. It just looks it's kind of silly, isn't it? Could I put it in my mouth? It's a good idea.
Creative solutions.
Yeah. All right. Thanks, everybody. Thanks for being here. Thanks, Karin, for that introduction. That was really sweet. Today, I'm super excited to present my master's research about different revegetation strategies for native wetland plant restoration in the face of Phragmites australis reinvasion and hydrologic extremes. Wetlands provide many services. They support biodiversity, they filter pollutants, moderate both floods and droughts, and they sequester carbon. And in total, these services add up to a lot of economic benefit. Inland wetlands alone give us over $27 trillion in services. However, historically, these wetlands have been viewed as wastelands, and this has led to the loss of over 20 to 50% of all of our wetlands globally. And those that remain are still at risk from land conversion or urban development, as well as further degradation. Now, plants support many of the functions and services. They're the primary producers, they form habitat, they stabilize soil, and they moderate those water fluctuations. But following disturbance events, these native plant communities do not always passively return. So in these degraded areas, wetland plant revegetation is a critical step to protect and restore these ecosystem functions. Yet wetland revegetation research has really lagged behind other ecosystems such as forests and grasslands. And this has led to a lack of established best revegetation practices. And it's still really unclear which methods will lead to the best native plant recovery. Additionally, wetlands face their own unique set of challenges, including from invasive species, because wetlands are particularly prone to invasions. And this is because they're situated downstream in the landscape, so they receive high amounts of invasive propagules, as well as excess nutrients, which favors invasive plant growth. And in wetlands, these invasive plants tend to form in these monotypic stands. So as you can see in this background image, these dense stands of invasive species can outcompete native plants and overall just reduce the habitat diversity. And one problematic invasive species is Phragmites australis. Phragmites is a widespread wetland invader across North America. It spreads clonally and via seed production. So although we have found these successful management strategies to manage those existing stands, reinvasion from seed germination is a reoccurring issue. And so it's even more important to establish these invasion-resistant native plant communities. And invasion resistance is just the ability of the native plant community to prevent or reduce invasive plant growth. And native plants can do this through diverse species interactions, where together they are occupying more habitat space and preventing that space for invasive plants to grow. And they can also do this through fast resource acquisition, where one competitive species can grow very quickly and preempt those resources from invasive species. Another challenge facing revegetation efforts is the early life stage bottleneck. And so for every amount of seeds that you're putting on the landscape, only a few of those seeds are emerging to become seedlings. And even fewer are persisting to adulthood to become fully established plants. And unfortunately, environmental stress events throughout these early life stages can further restrict that bottleneck. And this is because young plants don't yet have those adaptations to persist through these stressful environmental events. So in this case, there's even fewer seedlings that successfully emerge and persist to adulthood. And in wetlands, two key environmental factors that influence these community assembly processes are the hydrology and salinity of the site. So wetland vegetation communities form in accordance with the hydro period or the salinity gradient of that specific site. And while this is a normal process, we have seen shifting hydro periods due to these extreme weather events. And this can overall reduce revegetation success, because now all of a sudden you have seeds and seedlings that have mismatched tolerances with the current environmental reality. So in this thesis, using a field experiment in chapter 1, I investigate varied revegetation approaches. I sow functionally distinct native mixes, I vary their order and method of introduction. And then in chapter 2, using a greenhouse and mesocosm experiment, I test the effects of different hydrologic and salinity conditions on community assembly. And we test if a targeted species selection approach can be used to mitigate this hydrologic and salinity stress. So let's get into chapter 1. My study site was located in Great Salt Lake. For those of you joining via Zoom, Great Salt Lake is in the northern part of Utah. And specifically, my research was at Farmington Bay Waterfowl Management Area. And Great Salt Lake is a saline lake, and it's also a drying lake. And this is primarily due to upstream water diversions for agricultural and urban use. But it's also the result of these shifting arrival time and form of precipitation as a result of climate change. So whereas historically, most of our precipitation would fall as snowpack and persist longer throughout the summer, it's now arriving as rainfall and running off much quicker. And this has detrimental impacts to the wetland plant communities because there's reduced water to the Great Salt Lake communities, and these plants are experiencing more frequent drought conditions. And in 2022, in the first year of my field experiment, we actually recorded a record low water level at Great Salt Lake. Though, of course, I can't mention this record low water level without also mentioning our record-breaking snowpack, which occurred in 2023, in the second year of my experiment. And so this may lead some of you to ask, can we just rely on winter to refill Great Salt Lake and solve all of our problems? And the answer is probably not, because we would need 3 of those record-breaking snow years to refill Great Salt Lake and get back to that ecological sweet spot. In current revegetation efforts, managers typically introduce a suite of graminoid plants. And graminoids are just those grass-like plants you can see in that right-hand image. They provide habitat and forage sources for waterfowl and other wildlife, and they're also widely available. Managers can get those from the native plant nurseries in large quantities. Yet these species can struggle to compete against the fast-growing Phragmites, and so they don't always successfully establish in these revegetation efforts. And on the other hand are forb species. These are your broad-leafed plants, and they're just what you typically think of when you think of flowers. And they may provide more invasion-resistant properties. They have those broad leaves, so they're occupying greater habitat space. And some species are particularly fast-growing, so they can preempt those resources from invasive plants. But managers don't necessarily want to establish a forb-dominated field in these restoration sites because those forbs are not fulfilling all of their avian restoration goals. So one solution would be to sow forbs and graminoids together as a cover crop, because graminoids are the target restoration community and we still want to establish those. If we sow forbs with graminoids, the forbs will serve that invasion-resistant component and prevent Phragmites growth, but still allow that desired graminoid vegetation to establish. And in past experiments in wetlands investigating the use of cover crops, the cover crop did successfully reduce invasive plant establishment. However, it also reduced that desired vegetation, so it did not fulfill those restoration goals. However, in these experiments, the cover crop was sown at the same time as the desired vegetation. So one alternative would be to sow that cover crop at an initial planting date, and then later on, following senescence of that community, to come back in and sow your desired vegetation. Another consideration in revegetation efforts is whether to use seeds or seedlings. And seeds are often the preferred method, and that's because you have them in large quantities, it's highly scalable, you can use these big machines and cover a lot of the ground. However, there's often high mortality when we introduce seeds in revegetation efforts, and that's because of that early life stage bottleneck that we discussed earlier. We could also use seedlings, and I'll just call those plugs. And there's more upfront costs associated with using seedlings. You have to individually grow those out in greenhouses, and it's labor intensive. You have to hand plant each one. So you're covering less ground and it takes more time. However, there may be greater establishment success because you're essentially bypassing those field germination and emergence stages. And you're introducing those plants when they have more built-in resources through their root and shoot system. And so even though there's more upfront costs and labor associated with using plugs, it might still make sense for revegetation efforts if they have greater establishment. In this field experiment, we introduced two functionally distinct native mixes, that graminoid mix and the forb mix. We varied their order of introduction over 2 planting dates to get at that cover crop question. And we varied the method of introduction, either sowing them as seeds or planting them as seedlings using those plugs. And our predictions in this experiment were that plugs would increase native cover relative to seed introductions. Sowing forbs before introducing graminoids as a cover crop would increase native cover and invasion resistance. And that sowing forbs before planting graminoids as plugs would lead to the greatest native cover and invasion resistance because we expected those plugs to have higher establishment rates. And these are the species that were used in my graminoid mix. All of these species are currently in use around Great Salt Lake. These are the species we used in my forb mix. Forbs are not currently in use in current revegetation efforts because they're not yet widely available. However, we've investigated most of these species in past experiments in our lab, and some of them have shown really good promise. So now we'll just go through each of my treatments. So the first objective was those functionally distinct native mixes. We introduced a forb seed mix, which is denoted by the F, a graminoid seed mix by the G, or a combined forb and graminoid mix, which is our F and G treatment. We varied the order of introduction, where we introduced the forb seed mix at an initial planting date, and then a graminoid seed mix at a secondary planting date. So that's forb, then gram, FTG. We varied the method of introduction, where the graminoid mix was introduced either as seeds or as plugs, and the plug is just denoted by the P. So we have a forb, then graminoid as plugs, FTGP. And we also planted the graminoid plug mix with no initial planting, so that's just none, then graminoid plug. And last, we had an unplanted control just to monitor passive native recovery in the absence of any treatment. At Farmington Bay, we installed plots that were 1.5 meters by 1.5 meters, and we fenced them in for the initial portion just to prevent seed transfer. Our treatments were replicated 8 times, and they were arranged in blocks. Throughout the 2022 and 2023 monitoring seasons, we collected percent cover of individual species, and we also monitored the groundwater depth at each block via groundwater monitoring wells. To analyze this data, we used generalized linear mixed effects models, where our response was either native cover or invasive cover against the fixed effect of treatment and the random effect of block. And we'll just get into our results here. So on the x-axis are our different treatments, and I'll note that our control is on that far left-hand side. And the y-axis is total native percent cover in blue and our total invasive percent cover in red. And you may be able to tell that our treatment had no effect on either native or invasive cover. So compared to doing nothing in our unplanted control, there were no differences with any of our revegetation approaches in that native or invasive cover. And the big question is why, despite these varied revegetation approaches, did we not see any effect? And I think a large part has to do with the extreme weather events that persisted throughout my research. So what you're looking at is a drought monitor map from the first week of my initial planting in 2022. And as you can see, the majority of Utah was in extreme or severe drought. And then we had a lot of snow. And in that same week in 2023, we had no recorded drought at my field site. And this is what it looked like at my actual treatment plots. So on the x-axis here are the monitoring dates from 2022 through 2023. On the y-axis is the groundwater depth, and that horizontal black line represents our soil surface. So in 2022, the water was far below the soil surface. During our plantings, our treatment plots were just extremely dry, and this persisted throughout the whole summer. And in 2023, it was the complete opposite story. For the majority of the season, it was flooded, and it only briefly dropped below the soil surface at the end of the season. And you can see how these two conditions might favor different vegetation communities, and they occurred back to back with each other in this experiment. And so we did observe these species compositional trends between years, even though we did not see a treatment effect. So on our x-axis here are different treatments again, and on the y-axis is the average percent composition of individual species in each treatment for all of the recorded species that we saw. And in 2022, all of our treatments were dominated by 3 species: the alkali bulrush in that dark blue, saltgrass in the light blue, and Nuttall's alkali grass in the orange. And these species are drought-tolerant species. They're already used in Great Salt Lake revegetation efforts because of this. They require only short periods of inundation to fully establish. So overall, it's no surprise that they were the ones that dominated during our period of extreme drought. And then in 2023, we saw the emergence of the creeping spike rush and hardstem bulrush, which are both wetland obligate species, meaning that they require longer periods of inundation to successfully establish. So although we introduced all of these species in 2022, conditions were not quite right for these species to emerge in 2022, and it needed those flooded conditions to fully establish. So in this field experiment, extreme environmental conditions overwhelmed each of our treatments. And this really highlights the necessity to use bet hedging revegetation strategies for future restoration efforts. And this should include strategies such as sowing diverse revegetation mixes so that we can ensure that some species establish regardless of current environmental conditions. But it will likely have to be used in combination with other strategies such as repeated plantings and seedings and continued invasive plant management. And this recommendation really runs counter to the current emphasis on precision restoration in the literature. And this is the method that emphasizes putting the right seed at the right place at the right time. But this level of precision in revegetation is likely not feasible for many managers, given the increasing frequency and unpredictability of extreme weather events. So in chapter 1, we looked at these varied revegetation approaches, and we saw the impacts of weather on our community assembly outcomes. But we didn't necessarily explicitly test the effects of the environmental conditions on our community assembly. So in chapter 2, we aim to do just that. And I'll begin with the greenhouse experiment, where I test the effects of hydrology and salinity on community assembly. So water and salinity are tied together in semi-arid wetlands. As the water level goes down, the soil salinity increases. And this condition is really stressful for seeds and seedlings especially. Managers maintain coarse hydrologic control in these waterfowl management areas. So as part of their routine actions, they can release flushes of water, and this indirectly controls the soil salinity level as well. But overall, the salinity of these areas is generally unknown, and the interacting effects of the salinity and water condition on revegetation efforts is unclear. So in this greenhouse experiment, we used the same seed mixes as in our field experiment, that forb mix and graminoid mix. And we tested them at 2 water levels, 3 salinity levels, with and without Phragmites, and a Phragmites only control. Each treatment was replicated 4 times. We collected weekly percent cover of individual species, and we also performed an end of experiment biomass harvest. We predicted that with decreased water and increased salinities, plant growth would be reduced. So in general, at these increasing stress levels, plant growth would be reduced. We predicted that forbs would maintain greater cover and biomass than graminoids because we expected those forbs to grow more robustly than the graminoids. And last, we predicted that Phragmites would maintain greater cover and biomass than native plants under increased salinities. And this is just because Phragmites is known to be a salt-tolerant plant, and its salt tolerance is part of the reason it's been able to expand so rapidly throughout wetlands in North America. To create our water levels, we used a reservoir system to more accurately mimic a ground-fed wetland. We had a saturated water level where that reservoir was filled so that the water level was 2 centimeters below the soil surface. And we had a moist water level where the reservoir was filled 2 centimeters below the soil surface for an initial germination period, and then it was dropped to 8 centimeters for the remainder of the experiment. And we initially wanted to create more of a drought treatment. However, we just noticed that there was more water wicking, which was occurring throughout the soil column. So the differences between those two water conditions just ended up being less extreme. To create our salinity treatments, we added instant ocean to tap water and then mixed. So we had a fresh salinity level, which was just the straight tap water. We had a medium salinity level where we created a saline solution, which was 8 decisiemens per meter. And we initially filled all of our reservoirs with that saline solution and fully saturated the soil. And after that initial fill, we just refilled with tap water. And we had a high salinity level, which was 16 decisiemens per meter. And these levels were just based on our available soil analyses from the Great Salt Lake wetlands. To analyze this data, we used generalized linear models. For our native responses, this was native cover and native biomass against the fixed effects of water level, salinity, seed mix, which had 4 levels, the forbs with Phragmites, graminoids, and graminoids with Phragmites, as well as the interactions. And our Phragmites models looked very similar, except for seed mix had 3 levels, and these were the forbs with Phragmites, graminoids with Phragmites, and Phragmites by itself. We also measured the end of experiment soil salinity. So we created 1 part soil, 5 part distilled water solutions, and we converted those measurements to the electrical conductivity of a saturated paste. And these are the results from our end of experiment soil salinity. So we achieved a 4.8 decisiemens per meter level in our medium salinity level, and then 10.2 at our high salinity level. And this is roughly equivalent to about 2.5 parts per thousand at that medium level and 5.7 at the high level. Now let's get into our results. So we saw an effect of salinity, which varied by both seed mix as well as water level on our native biomass. And just for the purposes of today, I'm just going to be presenting biomass. On our x-axis are those different salinity levels, and on the y-axis is the cumulative native biomass. The forb seed mixes are represented in the purple, and the graminoids are in the green. And the first obvious result is that there were reductions in biomass at these increasing salinities. We saw that the forbs outperformed graminoids, and the difference between these mixes was pretty high at the fresh level, but declined with those increasing salinity levels. The forbs were also largely unaffected by the presence of Phragmites. So whether or not Phragmites was included in that mix or not, the forb growth was not shown to differ. And this is just to give you an idea of what this actually looked like in person. You can see particularly at that freshwater level, the forbs are outgrowing their bin. And then at those medium and high levels, the forbs start to look more similar with the graminoids mix. So now we'll look at our Phragmites response. And we saw an effect of salinity, which varied by both water level and seed mix. On the x-axis, again, are our salinity levels, and on the y-axis is now the Phragmites biomass. So first, Phragmites preferred saturated water under these fresh salinity levels. But at increasing salinity levels, there was no longer any difference between the Phragmites biomass at these different water levels. So this just shows that the increasing salinity levels are having more of a negative impact on Phragmites than that lower reduction in water level. Now, if we take a look at our seed mix responses, so the Phragmites sown by itself is in the red color. The Phragmites, when forbs were included, are in the orange, and then in the yellow with the graminoids. And we see that forbs greatly reduced Phragmites biomass under these fresh salinity levels. But forbs had no effect on Phragmites biomass at these increasing salinity levels. And now I'll just take a quick moment to talk about the graminoids with Phragmites mix. So as you can see from these two graphs, both the native cover and the Phragmites, or the native biomass and the Phragmites biomass for this particular mix was very low. And it was close to 0 grams regardless of treatment. And we suspect this is due to some methodological error which reduced seed viability in this particular mix during cold strat. So unfortunately, we were unable to determine the invasion resistance potential of the graminoids mix in this experiment. In this greenhouse experiment, the forbs outperformed graminoids, fresh and medium salinities, and they reduced Phragmites in the fresh salinity. And even though forbs had no effect on Phragmites biomass at those increasing salinity levels, the forb growth itself was unaffected by the presence of Phragmites. And this may point to the potential utility of including forbs in future revegetation efforts. Even if they can't prevent or reduce Phragmites reinvasion, we know we can at least establish some native cover which is itself resistant to the presence of Phragmites. In total, native plants and Phragmites were all greatly reduced at these high salinity levels. And so we saw that salinity does impact these community assembly outcomes and should be considered for future revegetation efforts. However, more than wanting to categorize these impacts, we wanted to see if we could mitigate some of these impacts from hydrologic and salinity stressors. So that's what we do in our mesocosm experiment. So the problem is we have these rapidly changing environmental conditions, and fine-scale monitoring or tracking of these water or salinity conditions for revegetation is often not feasible. So one potential solution would be to sow seed mixes that can perform well across a wide range of conditions. To do that, we created adaptive seed mixes. And within each of these seed mixes, we had species that were selected for diverging and broad environmental tolerances. And what that looks like, we had 2 adaptive mixes, and at the base of those were 5 core graminoid species. And then we added 4 forb species, and these differed between mixes. And each of those species were selected for diverging tolerances. So if we take a look at this golden dock mix, we can see further wetland indicator status. We have species that are facultative upland, meaning they prefer these drier sites, ranging all the way to obligate, meaning they require periods of inundation to successfully establish. And similarly, for salinity tolerance, we have plants that have no salinity tolerance, ranging all the way to high. So these are these 2 mixes, and they were just named for one species which had prolific growth in each of those mixes. So the golden dock mix, which is highlighted, and the sunflower mix, and the sunflower is highlighted. And our design was we tested these 2 adaptive native seed mixes at 2 water levels, 2 salinity levels, with and without Phragmites, plus a Phragmites only control. Each treatment was replicated 4 times and randomized. And again, we collected weekly % cover of individual species and performed an end of experiment biomass harvest. Our predictions in this experiment were that the adaptive mixes would maintain the same cover and biomass across all conditions. And this is because we expected differing species to perform well under any set of conditions. And we also predicted that Phragmites cover and biomass would decrease with increasing salinity and drought stress. To create our water levels, we used timed drip irrigation. We had a drought water level and a moist water level, and the moist was just watered for 5 times the amount of time as the drought. And these times changed as plant needs increased throughout the experiment, but that 1:5 ratio was always maintained. And I'll just note we had a really rainy June, so each one of our pools was fully saturated for the first 2 weeks of this experiment. We had a low and a medium salinity level, and because this was no longer a reservoir system, we just added salt directly to the soil. So we added 225 grams of instant ocean to whole mesocosms, and this was equivalent to our 8 decisiemens per meter solution, given the ratio of soil and water that we added. And we added 504 grams of instant ocean to our medium salinity level. For our native responses, we analyzed this data using generalized linear mixed effects models, where our response of native cover and native biomass was against our fixed effects of water level, salinity, seed mix, which was the sunflower or the golden dock mix, Phragmites with or without in their interactions, and the random effect of mesocosm because this was a split pool design. Our Phragmites response was analyzed using a generalized linear model. We did not have a random effect due to design limitations, so there was no split pool design for the Phragmites. But the seed mix had 3 levels, which was the sunflower with Phragmites, the golden dock with Phragmites, or the Phragmites by itself. And these are the results from our end of experiment soil salinity. So we used those same 1 to 5 solutions as in the greenhouse experiment. And we found that that end of experiment salinity was dependent on the water level. So whereas we did not record a difference between the low and medium salinity level under the drought treatment, we did see a difference in those salinity levels under the moist treatment. And we don't think that we didn't achieve any sort of salinity difference at the start of the experiment, but instead differing species growth between these 2 water levels just uptook different amounts of salt. So now we'll get into our native response. We saw independent main effects of water level, salinity, and seed mix on native biomass. And on the y-axis, again, is just that native biomass. But what we did not see was an effect of Phragmites. So whether Phragmites was included in the mix or not, native biomass was not shown to differ. And there were reductions in biomass at increasing stress levels. So moving from moist to drought or low to medium salinity, native biomass was reduced. We also saw that the sunflower mix outperformed the golden dock mix. And these results were driven by the presence of 2 dominant annual forb species. So again, here we're looking at an average percent composition graph. On the x-axis are different treatments. On the y-axis is the percent composition of all observed species. And when you can see the golden dock and the sunflower mix there. And both of these mixes were dominant by their namesake. In orange, the golden dock, and in the green-yellow, the sunflower. So now if we take a second and look at our Phragmites response, we saw an effect of salinity, which varied by water level, as well as water level, which varied by mix. On the y-axis, again, is our Phragmites biomass. And while there is no difference from the low to medium salinity level in Phragmites biomass, under combined stressors, medium salinity, and drought conditions, Phragmites biomass was reduced by over 50%. And if we look at our seed mix response, again, Phragmites sown by itself is in the darkest color. In general, drought reduced Phragmites regardless of seed mix. The inclusion of native plants in those mixes reduced Phragmites. And the sunflower mix was highly effective in reducing Phragmites, and the Phragmites biomass was close to 0 grams, regardless of water level. In this mesocosm experiment, we saw that the adaptive mixes were negatively affected by drought and salinity stress, which was contrary to our hypothesis. Yet, despite these negative impacts to the native biomass, these plants were still resistant to and reduced Phragmites across all conditions. And this may indicate that this targeted species selection approach could effectively be used to mitigate Phragmites reinvasion. Additionally, interacting environmental stressors negatively affected Phragmites, but not native plants. So native plants were only independently affected by either salinity or drought stress. And they may have some local adaptations, which helps them overcome these co-occurring stressors that commonly occur in our wetland systems. And although annual forbs drove our results, and some of the long-term implications of this data is still unclear, short-term benefits are still really desirable for managers because it reduces the cost and effort needed to manage those reinvasions. So looking at chapter 2 as a whole, some of my take-homes would be to seed in ideal environmental conditions when possible, because we saw those negative impacts of environmental stressors, even when species had wide-ranging tolerances. We should include forbs in revegetation mixes. In the greenhouse experiment, the forbs were resistant to the presence of Phragmites, and in the mesocosm experiment, the forbs reduced Phragmites under all treatment conditions. We should select for broad environmental tolerances to hedge our bets against future environmental conditions. But overall, we need to expect to use additional revegetation strategies on top of these strategies listed above. And I'll leave you with some management recommendations from my thesis as a whole. Yeah, I just have to emphasize this again. We really need to expect to use multiple and varied revegetation strategies. As we saw in my field experiment, there's just simply not a one-size-fits-all solution to successfully recover these native plant communities, given the complex interactions of reinvasion and unpredictable environmental conditions. But some strategies that I've gleaned from my second chapter would be to create those diverse revegetation mixes to help ensure that some species establish regardless of condition, and also to introduce fast-growing forbs to maximize our invasion resistance in these areas that have high reinvasion potential. So with that, I'd like to acknowledge my funding, which is from the EPA and the Ecology Center, and our project partners, the Utah Division of Forestry, Fire, and State Lands, and the Utah Division of Wildlife Resources. I'd like to say some thank yous, first and foremost to my advisor, Karin. I think Karin is such a strong advocate for her students' growth, and I'm really appreciative of all the time you've given me and to my growth in the past 2 years. Thank you to my committee members, Aaron Rivers and Tom Monaco, for the time and input that you've dedicated to me over the past 2 years. Thanks to Susan Durham for her statistical guidance in many one-on-one Zoom sessions. Thanks to Alec at the research greenhouses for his help setting up more greenhouse experiments than you saw in this presentation. Thank you to our project partners, David England and Keith Hambrecht. I'm really impressed with all the work you guys do to restore our wetlands in Great Salt Lake, and it's been wonderful working with you guys. Thank you to each and every member of the Wetland Ecology and Restoration Lab. I have to hand it to Karin on this one. She really chooses a great mix of people, and it's been really fun. A special shout out to Ray Robinson and Adrian Ernst, who were just really helpful in my first year forming my research questions. Thanks to my cohort members, Alana and Megan. It really would not have been the same without you guys. Thank you to all my family and friends. A lot of them are on Zoom right now. Hi. Especially thank you to my sister Grace and my brother-in-law Adam, who visited me twice here in Utah. And Grace is going to come help me move in a few weeks. Thanks to my partner Dre. And last, thank you to Maple. She was the MVP of writing my thesis. Thanks to my roommates for handling a 7-week-old puppy. And I want to give a shout out. All of my background photos were scans from the Intermountain Herbarium here on campus. I highly recommend their resources. Carl is right here in case you have any plant needs. And okay, I'll take any questions. Yeah.
Great talk. Do you have any way of comparing your results of your adaptive mixes to kind of more classic monoculture seed mixes or less diverse seed mixes to see if, even though they decline in the face of stress, do they decline as much as the other ones would?
Do you want to turn the speakers on?
It's on.
Okay.
Yeah. I think from my experiments, I don't have a way to do that direct comparison, but it would be very interesting to do from classic monocultures. My sense is that our classic monocultures that we're sowing do achieve a lot less cover and biomass just from like what I've seen in the field. And that's kind of why I've been pushing so hard on including these forbs in the mixes.
Thanks.
I know that there's not really a way that you can project what would have happened based on your experiment, but if the seed mix hadn't been compromised, what would you have expected the effect of the graminoid Phragmites mix to have been?
Well, we did still see the graminoids sown by itself had pretty low biomass and cover, and that seed mix was fine. It was just the graminoids with Phragmites seed mix. So I wouldn't expect it to be very invasion resistant. I think just the narrow growth habit that our graminoids have really struggle to compete against the broad growth habit of Phragmites. I think when they're fully established as adults, they have more of a shot, but at these beginning community stages, it's just really difficult.
With your two seed mixes where the golden dock and the sunflower were dominant, do you think that it would be beneficial to put those two species like in a mix together in the future or not?
Yeah, I'm kind of pro including all of these species, even if they're sown together. I think it'd be interesting to see like what might come out on top between those two. But golden dock is actually one of the ones that like is already pretty prolific across our field sites, and it's one of the ones that Alana saw throughout her field experiments. So that one definitely performs well. It'd be interesting to see the performance of the sunflower in these wetlands. Yeah.
Obviously beyond the scope of your experiment, do you have a feeling if you were to conjecture what the longer term impacts would be? Like it seems like the ones that take over really quickly, like the sunflower, the dock, they're all annuals. And
Yeah.
In year two, what would you see?
Yeah, I think it would really depend on the management actions. Like, I don't think we should be sowing these and then leaving the landscape to just recover naturally. I think if we start introducing these annual forb species, it really has to be combined with like a big push on continued invasive plant management to just come back in and take care of that Phragmites. So long term, if we don't do anything besides sowing forbs, I'm not sure we'd see a big recovery. Besides sowing the annual forbs at least. Any Zoom questions?
No questions so far in the Zoom. Lots of congratulations.
Hey, Peter, any questions? Okay, so no further questions for Maddie. Let's thank her for an awesome presentation.